Hey, Bio30s, and welcome to the start of Unit 6, Inheritance and Genetics. I like to call it classical genetics. Uh, if you're looking for this section in the textbook, it's going to be Chapter 17, pages 584 to page 621. In this section, we're going to kind of tie together the last four units that we've been discussing, starting back to cell division in molecular genetics and reproduction. What we're going to do now is we're going to look at the actual study of the traits that are basically being passed on by the bisexual reproduction through the formation of gametes, and what is sort of the end result of that, the actual traits that are getting passed down to the offspring through sexual reproduction. And that is the study of what we call genetics, which is the study of inherited traits. So that'll be our topic for this unit. I want to kind of do a little bit of an introduction, some kind of crazy science that, again, I like to discuss these early scientific theories because, boy, science is obviously an evolving discipline, and we don't have all the answers yet. And we certainly didn't have all the answers, a few hundred years ago. But the study of inheritance did sort of intrigue scientists for centuries. How did the offspring of a parent, perhaps, hey, I've got my mother's eyes and my father's ears. How did that actually happen? So scientists were intrigued by this for many centuries, and some interesting theories out there. Let's bring in Aristotle, because I love to bug Aristotle and his general stupidity. I've been bugging Aristotle. If you remember back to Science 9, I discussed that in Science 9. But Aristotle basically came up with a theory called pangenesis, which he suggested that the father's semen, which he considered purified blood, and the mother's menstrual blood actually interacted in the womb to actually create an organism's and influence an organism's early development. So that's what he thought, and that was what was causing certain traits that were coming from the mother and coming from the father. Leeuwenhoek, who I've talked about in Bio 20, who invented the microscope, the Dutch scientist who invented the microscope, he was the first one to see semen or sperm under a microscope, and he was convinced that when he saw the sperm under a microscope, that he literally saw a little mini human, and that was sort of the drawing that he drew right there. There was a little mini human that was sort of tucked into the head of the sperm, and he believed that it was the father who produced the human, and then it was just sort of incubated by the mother in her womb. So he believed that all the traits were coming from the father. And then I also talked a little bit about, well, I didn't talk about this gentleman, but de Graaf was another guy that came out with a theory that he proposed that the egg, not the sperm, contained the entire person. So he believed it was the female's egg that actually composed, that basically made the traits of that individual. It had nothing to do with the father's sperm. It was just a sperm that would help to stimulate the development of the egg. And then I discussed Leclerc last year in the evolutionary unit, and we discussed natural selection. Leclerc was one of the first scientists to propose a link between apes and humans, and that the Earth was considerably older than the Bible had actually predicted. And then he had to recant some of his views because the Catholic Church wasn't too happy with what he was saying. This was probably one that he should have recanted right off the get-go. He suggested that head and limbs came from the father and the rest of the body came from the mother. Don't even know what's going on there. But basically, by the 1800s, the running theory was that it was bits of the mother, bits of the father. You put them into a blender, and basically, if you had a yellow mother and a red father, your offspring would be orange. That was sort of the general consensus in the 1800s, until a monk came along by the name of Gregor Mendel, who was working in a monastery in Brno, which is now in the Czech Republic, but at the time was part of I believe it was the Austro-Hungarian Empire. But he was doing experiments in his monastery on pea plants, and that sort of became the basis for the study of genetics. And in his lifetime, he wasn't recognized. It wasn't until after his death that people kind of rediscovered his work and said, holy smokes, this guy was decades ahead of his time. And it's very interesting that he made all these discoveries because at the time he was doing his experiments, first of all, evolutionary theory had just sort of come out. Darwin had just proposed his theories of of natural selection. He was sort of doing his own thing with the study of genetics, which didn't really have a name yet. But neither one of these individuals realized how important their research was to the other. Obviously, now we use genetic, gene mapping and DNA analysis to see how closely related two species are to one another. But when Mendel did his work, first of all, we didn't even know what nucleic acids were. They hadn't been discovered yet. Miescher had not discovered nucleic acids at the time of a lot of Mendel's experiments. We had no idea the makeup of DNA, that DNA would pass down information. Right now, you've got more information about about DNA structure and how DNA works than Mendel ever did, because at the time of his experiments, DNA, people didn't know it even existed. So it was sort of a mystery. And he was able to basically formulate a theory about how genetic information was passed down without even knowing what was passing down. He just realized that something was doing it, but he worked out the mechanism beautifully without actually having a microscope, without doing any of that, the work that I discussed about in molecular genetics, or like what Watson and Crick did, like any of that. He was a century, in the past, before we even knew what DNA even looked like, and we didn't even know that DNA or nucleic acids even existed. So pretty incredible. This monk in this backward little town in the middle of the, what is now the Czech Republic, was able to do these experiments and actually come up with a theory that is still now considered to be extremely accurate. There was a few things that he got wrong, but he certainly laid down an amazing foundation, and that's why, for all intents and purposes, Gregor Mendel is now considered to be the father of modern genetics, even though he didn't realize he'd done it at the time. He did this by studying the traits of 28,000 pea plants. He grew and bred pea plants in his garden, and you can still go to the monastery. The monastery right here in the pictures. You can still go to Brno and see the monastery. You can go to the garden where Mendel tended his peas. It's sort of kind of a pilgrimage now for people that are, maybe into genetics, or that do genetics as a profession. It's sort of kind of, hallowed ground in the realm of, early genetic research. So what he did is he took pea plants that were true breeding. He took pea plants that he knew that after generation after generation after generation would always give him the same trait. So if he had a pure breeding purple flowered pea plant, it would always give rise, if you bred those two purebred purple pea plants together, they would always produce offspring that had purple flowers. So that's what's meant by true breeding. He didn't use hybrids, because if he would have used hybrids, then sometimes the purple flowered pea plants would give rise to white flowered pea plants, which was another trait. So he would always use pea plants that were true bred. He basically looked at 7 traits that were in the pea plants, and each of these traits only had 2 identifiable forms. So on this page here, you can see it right here. He looked at flower color, which was either purple or white, flower position, which was either on the end of the flower or on the axial branches. He would look at the seed color. They were either yellow or green. The seed shape, they were either round or wrinkled. He would do the pod shape of the peas, which were either inflated or constricted. He would do pod color, which was either green or yellow, or he would do the stem length of the pea plants. They were either going to be tall or dwarf. So he looked at 7 traits specific that only had 2 variations of each of the traits, and that's what he based his experiments on, was that those 7 traits, and those are the ones he was looking for. He, again, at the time, it was considered that if you had a purple and a white flower, you put them together, the offspring should be a lilac color. That would sort of be what the general consensus was among scientists in the 1800s. But what he discovered was something rather strange, that one of the traits would disappear. He would mix, the purple with the white, and then in the next generation, he would only find one of those 2 colors had shown up in the next generation. The generation after that, if he took those offspring from in the second generation and bred them, these are now hybrid crosses because they were coming from a purple and a white flowered parent. They had 2 parents. Each of them was a purebred. And then these were now all one color. In fact, they were all purple. He took those hybrid purple flowered plants and bred them. He found that the white trait would come back in the second, or in the next generation. So in generation 3, you would see basically the return of some of the traits. So he wasn't seeing a mixing of the color like he would expect what was the running theory at the time. So if you're interested in learning a little bit about Mendel's experiments, now would be the time to watch this video. This video will give you a much better view of the experiment that I can give you in front of this whiteboard. Please watch it. It's about 5 minutes, and you'll get a little animated view as to what Mendel did with his pea plant experiments. Then come back to me, and I'm going to finish up our discussion here about Mendel, and we'll finish up and wrap up our conversation today. All right, so if you watch that video now, you can see what he did. What Mendel found with these experiments was he would find that one of the traits, the purple, so I'm going to use the purple and the white flower example again because that's what's on this slide right here. He found that of those 7 traits, one of the, with the 2 variations, he found that so like flower color, which is purple and white, he found that one of those 2 traits would dominate over the other. So if I had purple and white and I mix them together, like a white flower parent and a purple flower parent, all the offspring in the next generation would be purple. So he believed that, okay, the one trait is dominating the other. So he would say that the purple trait was a dominant trait, and he said that the white flower trait was, in this case, a recessive trait because it was being masked if there was, if one of the parents had purple flowers, all the offspring would sort of lose that, they would lose their white coloration. There was no possibility of white coloration in the next generation. However, by the next generation after that, if he took organisms that had been bred but from those 2 purple and from the white and the purple flowers, in the next generation, if he had hybrid crosses that had a white and a purple parent, then he found that in the next generation after that, the white trait would reappear. So he found that this actually happened with amazing accuracy. He discovered that after he took that next generation, he found that there was a ratio that was very distinctive. He found that in the next generation, when the white floweration reappeared, he found that it was always in a ratio of 3 purple flowers to every 1 purple flower. And he did this with all the different traits. He did it with the seed shape, he did it with seed color, pod color, flower position, pod shape, and stem length. And you can see here in the results on this page, if you're looking right over here, you can see the results when he bred, thousands of plants. He got roughly always a 3-to-1 ratio. It was very close to 3-to-1, and it was always in favor of the dominant trait. So in the case of round and wrinkled, the round ones appeared more in the second crossing after they. So he would take a round and a wrinkled one that were purebred. He would put them together. All the offspring in the next generation at that point would be round. If he took those offspring and he bred them together, so he had round and round, what he found was those hybrid crosses, he would get, out of those hybrids, he would get 3 out of every 4 of the offspring would have a round shape to the pea pod or to the pea, and 1 out of 4 would have the wrinkled or the recessive trait. So that became very clear that there was a mathematical correlation here. There was very much something going on that was causing a ratio that was to be consistent. And this became the basis for Mendel's laws of inheritance. And the first one was his law of segregation. Now, this is going to sound very similar because we've discussed this already when it comes to the formation of gametes, to molecular genetics, to cell division. This is all stuff that we've sort of danced around with already. Here's the theory. I'm going to read it to you. He said that there were discrete factors that determined individual traits. He said that individuals had 2 copies of each of these factors, and when the gametes were formed, this pair of factors is separated, and the gametes receive only one of them. The factor that the gamete receives is determined randomly. The offspring then receive 2 factors of their own. One is provided from each of the parents, so they get two factors. One comes from each parent. Well, this sounds a lot like what? This sounds a lot like genes. Well, he didn't know what a gene was. He didn't know what a chromosome was. He didn't know what an allele was, but he's describing them right here in this law of segregation. He's basically laying out the formula for how genes and alleles work. So again, it wasn't until much later that the term gene was coined by a guy by the name of Johansen. He coined that. Mendel's factors became known as genes at that point. And then he said, hey, guess what? There's more than one version of a gene. We know that to be an allele. So this is nothing that's new. We've discussed this already here in Bio 30. But Mendel had basically laid out how genes and alleles worked before they had even been discovered. He just knew that there was some factor, something that was causing these ratios in the offspring, these factors, which we now know as genes. And if there's more than one type of a gene, and in the case of these, there are two types of traits. There's either like the purple or the white flower. There's a gene variation that causes the purple flower, and there's a gene variation that causes the white flower. So those are different alleles of the same gene, and the one allele is dominant to the other. So the purple allele in that case of his experiment would be the dominant allele, and it's basically dominating the recessive white allele, which is for the same gene. And that said, let me bring this back up to here. There are three combinations if you've got two copies of an allele. In the case of his experiments, there was only two variations of each trait, so there were only two alleles at play. There was a dominant allele and there was a recessive allele. Well, when we write these down in genetics, the dominant alleles are always written down with a capital letter. The recessive alleles are always written down with a small letter. This goes back to Science 9. This is stuff that you covered in Science 9. So if I have two copies of the dominant allele, I've got two copies of the same allele. So that is referred to as a homozygous configuration of the alleles. So I've got two of the same, homozygous dominant. I could also have two copies of the recessive allele. So that would be homozygous recessive, a little a. And those are both examples of what we call purebreds. So those would be the examples that he bred for his flower experiment. He would have had two purebreds, one that was homozygous dominant, the purple flowers, one that was homozygous recessive, the white flowers. And those are the two that he bred together to then get his hybrids. Now the hybrids would have gotten one allele from each of the parents. So all the offspring in that first mating would have been heterozygous or hybrids. Hetero meaning they've got one of each allele. Easy way to remember this is like homosexual, heterosexual. Homosexual, two people that are the same gender that love one another, and heterosexual means that there's two people from two different genders that actually love one another. So that's very, very easy to remember the designation of the names there. Homo means same and hetero means different. So those are the three types of allele combinations that we can deal with if we're dealing with essentially a single trait. And if we're dealing with a single trait is basically, if we're doing that kind of a cross, like what Mendel was doing, those are called monohybrid crosses. And in the case of his experiments, we had the actual physical trait of the flower, and I keep referring to the flower trait. The purple flower or the white flower refers to what's called the phenotype. The phenotype is the actual visible representation of the trait as what we can actually see. The combination of alleles that gets me my purple or white flower is known as the genotype. So the capital A, little a, or capital A, little a, those variations of the two alleles, that is known as my genotype. What it actually physically manifests as in the organism, the trait that it manifests as that we can actually see, that's the phenotype. So just so you know the difference between geno and phenotype. Well, how do I calculate all these numbers? Well, again, Mendel didn't have this because this wasn't invented until after him. But again, he was able to do his experiments. And we use now something very simple called the Punnett square, which was named after a real guy. There was a guy named Punnett. His name was Reginald Punnett, and he's the guy who came up with the actual Punnett square. You might remember your Punnett squares again back from grade 9 on grade 9 science when you covered this the last time. So how does a Punnett square work? Well, let's take a look at Mendel's experiment that he did, the one that I've been referring to throughout today's lesson, the whole purple versus the white flower purebred mix. So what he did is he took a purple purebred, and because it's a purple purebred, we know that purebred is always going to give purple flowers. Now, why is that? because it's got two copies of the same allele. It's got two dominant capital P's because it's the dominant trait in the flower of the pea plant. And then I've got a purebred white flower, so it's going to be homozygous recessive, and I'm going to represent that as little p in my Punnett square. So I got my capital P on the one side, little p on the top there. Now I'm just going to mix my two alleles together with each box. So when I'm done here, I'm going to wind up with all my offspring in the first cross between the purebred dominant and the purebred recessive, the purple and the white ones that are purebreds. They're all going to be, the ratio is essentially all going to be, it's going to be 1 to 0. There is really no ratio. It's essentially 100%. 100% of the offspring are going to be heterozygous. The genotype is capital P, little p. That's the genotype. 100% of them are all going to have that heterozygous configuration. Well, what does that mean for the phenotype? Well, because I've got one dominant and one recessive allele, the dominant allele is going to repress the recessive allele. I can't get a recessive phenotype unless I've got a homozygous recessive configuration. So in this case, even though I've got one copy of the recessive allele, the phenotype is still going to be dominant. So 100% of these organisms, or these plants, they're all going to be purple. So all of my offspring here are going to be purple after the first breeding. Now I'm going to take all of these offspring, or some of these offspring, and I'm going to mix them together in my next Punnett square. So the next Punnett square, I'm going to have essentially, these are going to be capital P, little p, and all of my parents in this Punnett square are going to be purple, but they're going to be hybrids, because these are purple, but they're heterozygous. They're still purple. The phenotype doesn't indicate what my genotype is, but in this case, I've got one copy of each allele, so that's going to have ramifications when I breed these organisms together. So what's going to happen? Well, I'm going to get a whole pile of combinations of alleles now. There's going to be a whole pile of combinations that are going to arise out of this. So what I'm going to wind up with now is I'm going to wind up with one of my offspring out of my 4 is going to be homozygous dominant. I'm going to wind up with 2 of my offspring out of that 4 that are going to be heterozygous dominant. And I'm going to wind up with one of my offspring in there that is going to wind up being homozygous recessive. So my genotype ratio now is going to be 1 to 2 to 1. One of them out of the 4 is going to be homozygous dominant. 2 of them are going to be heterozygous dominant. And one of them is going to be homozygous recessive. That's my genotype ratio. Is it going to be the same as my phenotype ratio, like what we saw up here, or my percentages? This would be essentially 25, 50, 25 would be the percentages here, right? Because 1 out of 4 is 25, whatever. Is my genotype ratio going to be the same? Am I going to have a 1 to 2 to 1 ratio? Well, I can't have a ratio of 3 because there's only 2 variations of the trait. It's either going to be purple or it's going to be white. So the question now becomes, which of these genotypes is going to give me the purple phenotype, and which of these genotypes is going to give me the white phenotype? Well, the only one that's going to give me the white phenotype is the one that's recessive homozygous recessive. This one here represents a white flower because it's got 2 copies of the recessive allele. The other 3 offspring, even though they're different genotypes, they're all going to have the same phenotype, which is purple. So in this case, my phenotype ratio becomes 3 to 1. Wait a second. That was the ratio that I just mentioned that Mendel kept finding in his research. Without the use of Punnett squares, he just recorded the offspring and what color they were after each of the breeds, well, after the breeding incidents. And guess what? There's my 3 to 1 ratio that's very common. This is an interesting one to keep in mind. This is one that I would keep in mind. Every time you've got a 2 heterozygotes that come together in a monohybrid cross like this, 2 heterozygotes is always going to give you these ratios all the time. These ratios are pretty much set in stone. When you have 2 heterozygotes that are mixed together, you're going to have a 1 to 2 to 1 ratio genotype-wise. That's going to be 1 homozygous dominant to 2 homozygous recessive, to 2 heterozygous. Let me do that again. 1 to 2 to 1. One of them is going to be homozygous dominant. 2 of them are going to be heterozygous dominant, and one of them is going to be homozygous recessive. At the same time, though, The 1 and the 2 are going to be represented as a dominant phenotype. So the 1 plus 2 becomes 3 in my phenotype ratio. I'm going to have 3 of the offspring out of 4 that are going to be purple flowers. And in this case, 1 out of the 4 is going to be the white flower. And again, everything I just described to you is on the bottom of this slide. If you haven't seen it already, it's all there. I just ran you the Punnett squares on the top. Okay, one last thing to finish up with and we're done for today. So here's the question. How do I know if I've got a purple flower? Let's say I got purple flowers and I got purple flowers on my pea plants. How do I know if that purple flower is coming from an organism that has a heterozygous genotype or a homozygous genotype? How would I know that? Well, what I need to do is I need to do a test cross. So what a test cross is the phenotype is dominant, but I don't know whether it's a heterozygous or a homozygous genotype. This could have real ramifications. For example, on the slide here, I've got an example of sheep. Now in sheep, black wool is not considered to be desirable in sheep. Black sheep, when they have black wool, it's very brittle and it's very difficult to dye. Hence the term, you're the black sheep of the family, right? So what ranchers want to do with their white sheep is they want to have purebred white sheep because they don't want white sheep in there that are heterozygous. They might have one copy of the recessive allele for black wool, and then 2 of them come together, and then you got a 1 in 4 chance of having offspring that are black sheep. So these farmers, these ranchers, can figure out which ones are purebred by doing a test cross. And a test cross always involves the organism that's the unknown dominant genotype. We're going to breed it always with a recessive organism. Now, why is that? Well, a recessive organism, if I know that it's black sheep, I also know that I know what its genotype is. There's only one possible genotype for a black sheep. It's going to be homozygous recessive. So if I know, if I have a recessive organism, if I know the phenotype, I also know the genotype. If it's a dominant organism or a dominant trait, I know the phenotype, but I don't necessarily know what the genotype is because it could either be homo or heterozygous. So how does this work? Well, I've got 2 scenarios for you here. What would happen if I had an organism that was heterozygous white? So it's got 1 copy of the white allele, which is dominant to the black allele, and I'm going to breed it with a black sheep. And then I've got a purebred white sheep, and I'm going to breed it with one of these black sheep. Run the Punnett square right now. Pause me right now. See if you can run the Punnett square numbers to see what happens. And then I'm going to run you through it really quickly just to see if you're on the right track or not. Okay, here's what's going to happen. Well, if I breed my white sheep with my heterozygous white sheep, the one that's a hybrid, here's what's going to happen. I'm going to have a ratio of 1 to 1. The offspring that are going to result from this pairing, half of them are going to be white, half of them are going to be black. So if I see any black sheep coming out of the mating in a test cross between an unknown dominant genotype with a recessive organism, and if the recessive trait comes through, then I know I don't have a purebred animal. If I had a purebred animal, here's what happens. A purebred animal is going to be like Mr. Mendels Experiments, where you're going to get all of your offspring that are going to be hybrids. However, they're all going to have at least 1 copy of the dominant allele, which means that all the offspring of that crossing are going to be dominant. There will be no recessive organisms in that test cross. So if I see that pattern occur, then I know that my dominant organism that I just bred, I did the test cross on, it's got to be a purebred. It can't be a hybrid organism. That's an organism that I want to keep in my breeding pool because it's only got the dominant alleles handy for me. That said, is it easier to keep a purebred dominant herd or a purebred recessive herd? It's much easier to keep a purebred recessive herd because, again, if you know the phenotype, you automatically know that it's got to be homozygous recessive if it's a recessive trait. So recessive herds are actually easier to maintain. If you see a dominant trait show up in the recessive herd, then you just don't let that animal breed, and that will prevent that dominant allele from being passed down to other members of that herd or whatever. Um, and one last one to finish you off on. Here's the if it only worked this way, but it does. Sometimes the alleles are not perfectly dominant over other alleles. That's what my discussion will be next time. It'll be on co and incomplete dominance, which I'll be discussing with you next day. There are some alleles, some genes that don't work on a complete dominance scenario, which is what I've been discussing today. It's been all about complete dominance. There are some situations where genes are only codominant or incompletely dominant over one another, and then you get spotting or you get mixing of color, which I will get to next day. For today, though, to kind of wrap up your knowledge about today's lesson, I would like you to complete the workbooklet, a monohybrid and test crosses. Lots of good Punnett square questions in there for you. Run your head through that booklet, see what you can figure out from there, and we'll be discussing any questions you have on that when you get to class next day. That's it for today. Thanks for tuning in, and we will see you very shortly as I discuss my next topic in classical genetics.